So hello everyone. I will just start with a few slides about the 4D-BGC SCOR working group that organized this 4D-BGC webinar series. So today we are glad to hear about the work of Nicolas Mayot. So I co-chair the group with John Sharp, that is here online with us. Our group is about the coordination of the development of gridded 4-dimensional data products from BGC Argo observation. So the main mission of the group is to facilitate discussion and coordination among different scientific communities around the development, the validation, and the distribution of 4D BGC products from observational datasets, and with a specific focus on the BGC Argo array. So the goal of this initiative is to significantly enhance access and utility of BGC Argo observations through the 4D BGC products, and thus refine our understanding of ocean biogeochemistry, improve models and reanalysis products, and inform policy decisions. So you can see the picture of the group that had a first meeting during February 2024 during OSM, and we will have our second one next week in Vienna during EGU. So we are very pleased to say that the BGC Argo-based data products webpage is online through the BGC Argo website. So it's a catalog with all the BGC Argo data products that are available for now with specific information and the links that could interest you. So you have here an example of one product. Here you have the web page of the SCOR working group. So we are giving the information about the webinar series, the group information, group activity, and resources on this website. So you can have a look and see what is going on here. And don't forget to sign up to receive updates on the 4D BGC activities, to sign up the mailing list. And you can also find all the previous webinars on the YouTube 4D BGC webpage. And so now we can hear Nicolas Mayot presentation. So I will stop sharing my screen. And you can share yours, Nicolas.
Yeah, I'm going to share mine. Can you see my screen?
Yes.
Is it full mode?
We can see the... No, it's okay now.
Okay.
Thank you very much.
Thank you. Thank you for the invitation, for giving me the time to talk about my research. So, hi everyone, I'm Nicolas, or Nico Mayot. I'm going today to talk about phytoplankton phenology in the Southern Ocean. It's a study that I've done with some colleagues here in Villefranche, so Raphaël, that just talked, and also with Julia Witz, and also with some colleagues from Marseille in France, David and Lloyd. And so I will start first by showing you a bit about my career path, just to let you know who I am and where I come from. And after I will start to present my scientific results. So in terms of my career, so I did a PhD in Villefranche-sur-Mer already with Fabrizio D'Ortenzio. At that time, I was studying the Mediterranean Sea, but I was already using some BGC Argo data. After my PhD, I've done a first postdoc in the US, in Maine, at the Bigelow Laboratory for Ocean Sciences, in the research group of Pattimatry, and I was working at that time on the Arctic Ocean, also with BGC Argo float. And then after 2.5 years, I joined at the University of East Anglia for a second postdoc in the research group of Corinne Lequeré, so in England. And it's at that time that I started to study the Southern Ocean. And I was also more focusing this time on the ocean carbon sink. And so I'm going to explain it a bit after. And then, since last year, I came back to Villefranche for a third postdoc with Julia Witz to work on the Southern Ocean and mostly more on the phytoplankton phenology that I'm going to present. And I also obtained a CNES, so a fellowship from the CNES, so the French Space Agency, to support, that is going to support this work for the next 2 years. So in terms of my research, or what I'm going to present today, so about the Southern Ocean, why is it important to study the Southern Ocean? To explain this, I'm going here to use a figure from the Global Carbon Budget, which is every year publishing information about the CO2 emission and the CO2 sink. And so on this figure, which represents on the y-axis the CO2 fluxes, and on the x-axis the time, so the year. The positive side of this figure is representing the CO2 emission. And you can see that most of this emission, of course, come from the burning of fossil fuel. Another part comes from the land use change. And this is so inducing some CO2 emission. And this CO2 that have been emitted, where is it going after? Some of this CO2 is being absorbed by the ocean. That's what you call the ocean CO2 sink. Another part is being absorbed by the land biosphere. And all the CO2 that have not been absorbed by the ocean or the land end up in the atmosphere, where it accumulates and induce climate change. So we can see clearly on this figure that the ocean is an important part of the global carbon cycle, because it absorbs every year around 25% of the CO2 that have been emitted. And the Southern Ocean by itself corresponds to 50% of this ocean CO2 sink. So that's why it's very important to study the Southern Ocean and get more information on the ocean CO2 sink. And so now, how can the 4D-BGC, so biochemical data product, be used in this context of the global carbon budget? And to explain this, I'm again using here a figure from the global carbon budget, which represents on the y-axis the CO2 flux, and on the x-axis the time. But this time, I just want to notice that positive values mean carbon going, or CO2 going into the ocean. And so in the global carbon budget, there is two methods that is being used to estimate the amount of CO2 which is being absorbed by the ocean. A first method uses simulation from global ocean biochemistry model. So it's represented here in blue. The thick blue line represents the average of all models, and the thin line represents every individual models. The second method, which is being used by the global carbon budget to estimate the ocean carbon sink, corresponds to the use of what you call database products, which are 3D gridded data product for surface partial pressure of CO2, so PCO2. So they use data from the SOCAT database, for who's that knows what it means. And though they interpolate this database to produce this 3D gridded data product. So we can kind of say that they are analogous to the 4D-BGC data that we are talking in this webinar, in the means that they also use machine learning methods to interpolate observational gap. And to come back to the ocean CO2 sink, in the global carbon budget, because we don't know who is right between the database product or the global ocean model, what they do is that they make an average of these two estimates, which is represented here by the black line. So it's what I was to mean here. So the ocean CO2 sink that the global carbon budget is reporting is the average of these two estimates. And so how can the 4D-BGC data can be used to inform, or like to explore, why are there some differences between these estimates, and how can we know more about the ocean CO2 sink? One way is to use some 4D-BGC data that can be directly related to the ocean CO2 flux. For example, I'm thinking here about 4D-BGC product that should, for example, interpolate dissolved inorganic carbon data. And so from this 4D database, you can estimate the exchange of CO2 from between the atmosphere and the ocean. Another way to use the 4D-BGC data to better inform the ocean CO2 sink is that this data product can provide an observational constraint for the model that are being used. So it's like they can provide a benchmark toward which the model should try to get. And so it's more in this context that my study is related to. And in this sense, why is it important to study the Southern Ocean phytoplankton phenology? To explain this, I'm using here this figure, which represents the seasonal ocean CO2 sink, simulated by the model in green and estimated by the database product in blue. And notice that this time the ocean CO2 sink is represented by negative value. Sorry, it's always changing. For the winter season on the right and the summer season on the left. And what you can see here is that during the winter season, all models kind of agree together. They are kind of a consensus about the value. But in summer, there is a high intermodal variability in terms of the CO2 which is being absorbed by the Southern Ocean. But during this season, the summer season, is during this season that the phytoplankton is blooming and is helping the ocean to absorb CO2 because it's performing the photosynthesis, which is absorbing CO2 to produce carbon biomass, and so help to reduce the surface concentration of CO2 in the ocean, and so to help to the ocean to absorb CO2. And so we can see that in summer, for the ocean models, there is a lack of consensus about the impact that the phytoplankton have on the CO2 fluxes. And so that's why it's important to provide more information about mostly the timing of the bloom, the amount of production which is happening, the viability, etc. And so to help or to provide some of this benchmark, we propose here, the objective of this study is to propose to study the phytoplankton phenology by producing a regionalization of the Southern Ocean, which reproduce the spatial and temporal variability of the phytoplankton phenology based on the 4D-BGC data which is being produced. And then to study the variable, the environmental variable that influence this regionalization. And so to do that, I'm going to continue. So the data which is being used, so first is supported by the BGC, or the Biogeochemical Argo program, which is associated with the Argo program, which is an international program for deploying Argo floats, where you have here a picture of an Argo float, which is a robot, which is measuring mostly, firstly, salinity, temperature, and pressure with a CTD, and is performing this profile every 10 days. And during the remaining days, it's drifting at depth. And so every 10 days, the robot wakes up and makes a profile and transmits the data via satellite to laboratories. And within this Argo program, so the BGC Argo program, with some of this float being equipped with additional sensor to measure BGC variables. And there are mostly 6 variables which is being measured, which are oxygen, nitrate, chlorophyll, BBP, remember pH, and yeah, that's it. And irradiance, and that's 6. But here I'm going to focus on 2 of these variables, chlorophyll and BBP. And actually, right now, there is around 500 of these floats that are being deployed or being active in the ocean. And the target is to increase this array with around 1,000 of BGC Argo floats for the next decade. And so we can use this data, so the chlorophyll and the BBP data profile, to estimate the phytoplankton biomass and also the amount of particulate organic carbon and also the phytoplankton carbon biomass through some kind of relationship. So with this data that are at the base of the 4D BGC data that I'm using. And I have to mention here that the Southern Ocean has been particularly, sampled with this Argo float through a lot of several important projects, namely the SOCOM, the Rainbow Sail and SOCLIM project, for example. And so this provides a good database to get some information in this Southern Ocean. And so the 4D BGC data that I'm using come from the work of Raphael. So I'm going to try to explain, but if I'm making some mistakes, I'm sorry already, Raphael, you can correct me after. So this database are based on a neural network that have been trained with BGC Argo data, and which is matching up satellite information with TS information from Argo float to reproduce at the end a vertical profile of chlorophyll and BBP. And so this neural network is based on the multi-layer perceptron. I'm not sure about the pronunciation, but I think it's this. And these three main components of this neural network. The first one is the satellite information at the surface. So here using ocean colored information. Match up with TS information from Argo floats and also geolocalization. And at the end, after training, this neural network is able to produce a vertical profile of BBP and chlorophyll. And so to explain, to show you the training, so you can see that here, for example, so this neural network has been made to reproduce the global ocean, but here, because I'm focusing on the Southern Ocean, I'm just going to show you the database available for the Southern Ocean. So you can see that we have a lot of data available to do some training and validation. And there's also here, you can see in green, an independent data set that Raphael used to validate this 4D-BGC data product. And I'm showing here the time series from this independent data set from the BGC Argo program versus what the neural network is producing in terms of chlorophyll. And so we can see that it's able to reproduce both the vertical distribution as well as the temporal variability. And so now by giving to this neural network a 4D data set of TS data, we can have a 4D data set of chlorophyll and BBP. And the spatial temporal resolution of this data set is a quarter of a degree between 0 and 1,000 meters. And for 24 years, from 1998 until 2022, at a weekly time resolution. And this data set is freely available through the Copernicus Marine System Marine Services. So this is the data that I'm using to produce a regionalization on the Southern. So it's a global data set, but I'm here just subsetting the Southern Ocean side. And the method that I'm using to produce a regionalization is based on a clustering method. And I'm going to show you which one and how I'm doing this. So first, from this data set, so the neural network data set, what I obtain is for each week and each year, I have 2 cubes for like BBP and chlorophyll, so longitude and latitude and versus depth. So it's like a 3D cube for each week. And I have this for each week and each year. So that's what we call a 4D data set. And first, what I'm doing is that I'm doing a weekly climatology. So I remove the information about the year. So every year, week 1 of every year is being averaged together. So I'm here producing a weekly climatology of BBP and chlorophyll profile. Then, before to give this information to the clustering method, I'm going to use some principal component analysis to simplify the information. So I'm going from this 4D data set to a 2D table. But this 2D table has, so for each row represents a week and a pixel, and the column represents the data in terms of profile, so a BBP profile and a chlorophyll profile for each pixel for week 1, for each pixel for week 2, for each pixel for week 3, etc. until 26 weeks, because I'm not using the full 52 weeks because there are some a month without data because we have no information through satellite images because of a night in the austral area. And I'm also using only the information between 0 and 100 meters. Then I'm performing firstly a first principal component analysis to try to synthesize information about the profile. So for now, for each pixel and each week, I have 3 principal components, so 3 variables. Then I'm restructuring this table again to this time having the information through each pixel and for each principal component. So it's kind of represent time series of principal component for each pixel, but this information is still quite important. So that's why I'm performing a second principal component analysis. And so now I end up with a table with information for each row represent 1 pixel, and each column represents a principal component axis, or principal component axis, yeah. Principal components. And this table that I'm giving to the clustering method, which is a Gaussian mixed model. And I'm choosing a number of groups. For example, here I have choose 6 groups to, at the end, give me a map of the Southern Ocean. So that's what I'm calling the regionalization of the Southern Ocean based on the phytoplankton phenology, which is represented by this information from chlorophyll and BBP. So I hope it's clear enough for everyone. So I've done this in terms of climatology, but I have also done it for each year. So this time, I'm not doing any averaging, but I'm just extracting the information for each year. And instead of redoing the full process, I'm just projecting this information for 1 year into the principal component analysis that I have done before, and also apply the training method that I have, the clustering as applying the trained clustering method that I've used before to produce a map for each year. So if I try to synthesize this information all, I have 1 map, which is a climatological map of the Southern Ocean, and I also have 1 map for each year, which allowed me to do some study about the internal variability. And so I'm going now to present some result out of this method. So here is the map of the Southern Ocean, and based on the phytoplankton phenology. So here, each pixel which shares the same color, so each region represented by 1 color. So each pixel within this region should have a similar variation in terms of vertical profile of chlorophyll and BBP over time, over a year. And so, for example, for this region in brown here, I represent in this figure here the time series for chlorophyll in green. So from 0 to 300 meters for over a year. Here for BBP in orange. And the third graph here represents the vertical integration of BBP that has been converted into a carbon associated with phytoplankton based on some relationship. So I've done this this is for this brown region here, but I have done this for all of the 6 regions that is represented on this map here. And you can see that you have some spatial and temporal variability, which are different from one region to another. And so based on this information here, on this time series, and based on the location of this region, you can see that it's different over the latitude, we can make already some kind of interpretation of what does this, or what are the information behind this this region. So the 2 first regions here could be related to what we call subtropical typical variability, because they mostly are located in the low latitude and also have some kind of deep chlorophyll and deep BBP information at depth over the summer season, which is typical of subtropical region. The area located closer to the Antarctic are associated with a higher concentration of chlorophyll and BBP. And you can see it also here in terms of carbon related to phytoplankton, because the y-axis here for this region have a different unit compared to the one before. So there's more biomass associated with this region. That's why these 2 regions are mostly we are choosing to relate them to the polar and the Antarctic. And these two regions in between the polar and the Antarctic and the subtropical, so the yellow and the light blue we decide to call them the subantarctic region. So that's it for kind of a general interpretation of this regionalization. And as I have said before, I have done this regionalization for each year. So you can imagine that you have this map here, but for every year, which allowed me to do some analysis about the long-term viability and the internal viability. And what I have done here first is that I look at the area associated with each of these regions and see if over time I have one region which is increasing in terms of the size or decreasing. And in fact, when I look at this area for each region over the last 20 years, I don't really see some high trend in terms of variation of the size of each region. We can maybe argue that the region here, the yellow one, which is related here in the Pacific region, could have an area which is decreasing over time, but that's it. Like I don't see big changes in terms of the size of each region. And to confirm this, what I have done here is that I have done the average of all the maps for the first decade versus the average map for the second decade. So we can compare like a decadal variation of the map. And when you compare these two maps here for the first and second decade, you cannot really see a clear difference. It's like, it looks like the regionalization that I propose as a climatological state is kind of stable over time. So that's my conclusion here, is that the regionalization that I have produced is relatively stable over the last 20 years. But I don't see a large viability over time, like internal or decentral viability. And to go further, so this was the map that I obtained at the climatological level. I have also looked for each pixel, how each pixel vary over time. So I ask, by looking at all of this map here that have the 24 maps here, I try to, for each pixel, to select which region is showing up the most of the time. And on this map here, I'm only representing the pixel in color for which it's showing the same region 75% of the time. So it's like regions that are very stable, but if for every year, if you take one pixel, you have 75% of chance to have this color, which is appearing. So that's what I call here the core region, like the region that are not changing that much over time. And what I see is that these core regions are being separated by some borders here, where there's like a transition zone, where the regions are changing a lot between one year to another. And so it looks like that we have clear border between each region, which are like also stable over time. So we have core region and border that are like kind of stable each year. And so now I try to understand, why do I have such strong border between them? And first, we can hear what I'm showing is the location of the front, of the subantarctic front, so a front is associated with the Antarctical circumpolar current. And we can clearly see that this front is related to the border between this subantarctic region and the polar region here. And in the past, in previous publications, it has been shown that this front, which is occurring a lot, so in this area here, is also kind of a border for the biology. And so that's why it's kind of shaping the location of my region, is because it's a strong physical processes or theater. A second borders is the one between the subtropical and the subantarctic region. And these borders here can be related to the differences between the area which is nitrate limited versus the area which is iron limited. And to locate the the border between these two different biogeochemical provinces, I use data from the World Ocean Atlas for the nitrate. So a data set that have not been used for my clustering or even for the neural network that Raphael have been using to produce a 4D data set. And so I'm representing here the time series of a nitrate associated with 3 of these regions, so the brownish one and the yellow one here. And you can clearly see that for the first 2 regions, the subtropical region here, we have some very low nitrate in summer, and which is increasing a bit in winter in this region, in the light brown one. But in the yellow one here, we have nitrate almost all year round. So this area is not nitrate limited. From this, southern of this line here, the dotted line here, we are not anymore nitrate limited, so we are probably iron limited, which is well known for the Southern Ocean. And so we can now see that the border of my region are related to physics and nitrate availability. And so this is shaping as well the phytoplankton phenology. So if you want to reproduce the phytoplankton phenology in a global ocean model, you have to be able to represent this physical and biochemical features. So now, because we have information about what's going on at depth, we can also study some processes going on at depth, and notably the appearance of what we call the deep chlorophyll maximum. So sometime in the Southern Ocean, and mostly during the summertime, we have what we call a deep chlorophyll maximum at depth, which is occurring. And this is because the phytoplankton have to adapt to the fact that there is no more iron available in the surface layer, but there is more at depth. And so the phytoplankton is increasing, or is either increasing the phytoplankton, is chlorophyll content to be able to harvest the low light available here, or is growing at depth, so there's a real increase in terms of biomass, like there's more phytoplankton cell at depth to be able to harvest both the light and the nitrate and iron available in subsurface. And so from the 4D dataset of chlorophyll, I can study the occurrence of this feature here of a deep chlorophyll maximum. And this is shown here on this map. So the area that are the greener are the area where you have the most of the time you have a deep chlorophyll maximum occurring. So it's mostly observed in the subtropical region, but you also have some of this feature in the polar area, and mostly here south of Australia. So in the real HNLC, or like high nutrient, low chlorophyll area. And as I said, you can also, because we have also the BBP information, we can also see if we have a real increase in terms of biomass. So if you have a real increase in terms of carbon content at depth by looking at the profile of BBP. And if you observe an increase in the BBP at depth, we can call this what we call a deep biomass maximum. And so from the 4D dataset of BBP, I can look also at the occurrence of these features here. And you can also see that it's occurring mostly in the subtropical region, less occurrence than the chlorophyll as expected, but you also see them here in the HNLC region. So I go a bit more into detail about these features, and for this I'm going to focus on the 4 regions where we observe those features, which are mostly the one associated with the subtropic, the yellow one here, and the HNLC, or the polar region, the light green one. And so these figures here is representing over the year the occurrence of the deep chlorophyll maximum and deep biomass maximum for the 4 regions. So the first 2, the second one, and the fourth one. The continuous line is representing the DCM, so the chlorophyll, and the dashed line represents the deep biomass maximum, so the DBM. And so here, when you have like 100%, that means that every pixel of this region is showing a DCM or a DBM and or a DBM. And if you are like at 0, that means that none of the pixel associated with this region is showing this type of features. And so we can see, for example, that in the polar region, this feature mostly appear in January and in February, so in summer, and only for 30 or 40% of this region. And we can also look now at where, in terms of the vertical distribution of the DCM and the DBM, and relate them to the vertical variability of the mixed layer depth. And we can see that we have differences with, between the DBM and the DCM. We have a DBM which is occurring at shallower than the DCM. And also that there's a clear differences between the mixed layer depth and these features for the subtropical region compared to in the polar region, which is like where the mixed layer depth and the DCM and DBM is really closely related to. And to go a bit more into detail, what I've done here is that I look at the month of January and I make up the average of all the profile that are showing this type of features for each region. And I'm showing here, so for the x-axis, the chlorophyll and BBP being dashed line and chlorophyll being the continuous line. And the black line is representing the mixed layer depth. And you can clearly see what I was saying is that here the DCM and the DBM are really far away from the mixed layer depth. So it's a very deep DCM and DBM. While in the polar region, the DCM and DBM are really related to the mixed layer depth. So it's really an accumulation of phytoplankton very at the base of the mixed layer depth. So that's why what you can do is like an example of what you can do when you have such a 4D data set. So now, as I said before, I have been able to study the internal variability of some of this region, and I try to understand this internal variability of this region. And for example, here, for the last part of my presentation, I'm going to focus on the HNLC region. And this region, the hypothesis is that the phytoplankton production in spring is occur because in winter we have a strong vertical mixing which supply iron for the surface layer, and so allows the phytoplankton to grow during the spring and summer. And so here I took the information in what I call the core region of this HNLC region, and I compare on this graph for each pixel the relation between the winter mixed layer depth, so the mixed layer depth that occur in June, July, and August, versus the phytoplankton accumulation in spring. So it's like the differences during, like it's the accumulation between the max of biomass occurring in spring versus when it starts to increase, when the blooms start, kind of. And so here I just present like when it is like more yellowish, it's because there's a lot of points, so a lot of pixels. And so we can clearly see that this kind of relationship between the mixed layer depth in winter versus the biomass accumulation in spring, and these relations. So like here it represents the Spearman cross correlation, which is represented here. So it's quite it's like moderate, but yeah, this can see by high that there's a relationship going on. And this is showing also what I call the spatial variability, because here I'm using climatological data. So I'm just making the average of every point every year in this region. Then what I have done is that I have represent the winter mixed layer depth and the biomass accumulation average for every year. So each point here represent 1 year. So we can now, I could have said here that in fact I should have maybe changed this to the anomalies, like the winter mixed layer depth anomaly, annual anomaly, versus the annual anomaly of biomass accumulation. And what we can see is that kind of have a relation between when the mixture depth is deeper for 1 year, you have a higher chance to have a higher biomass accumulation versus when you have a year where the mixture depth is being shallower. And to confirm this, what I've done is that I make the average of every time series for this year here that is inside this red circle versus every time series of phytoplankton biomass in this year, which is in a blue circle. So this is these 2 time series here over time. So in biomass of carbon, so in gram of carbon per meter square, so coming from the BBP data. And so if you have a deeper winter mixture depth, you can expect that you are going to have a higher and a delayed accumulation of phytoplankton biomass versus if you have a mixture depth that have been shallower in winter, which is represented here, so in blue. Then I took again this graph here and I look at the SAM index, which is a southern annular mode, which is a climatic index in the southern ocean area, which mostly related to the wind speed and the wind intensity. And so when you have more positive index, that means that you have higher chance to have a year with a strong wind versus a year where you have a more negative value. That means that you have a higher chance to have a lower wind. And it kind of looks like that when you have a SAM index which is more positive, you are mostly going to have a deeper winter mixture depth and a higher biomass accumulation in the HNLC region versus when you have a year with a negative index, SAM index. So SAM is probably one of the environmental factors that explain the internal variability of the phytoplankton phenology in this region. And so to finish quickly and to have some time for the question, I'm just showing you some perspective of this work. So we would like also to apply a primary production model to this 4D data set to be able to translate this information on the phytoplankton biomass into global, into gram of carbon per meter square per day, so production, and by using also a 4D data set of light. And we also would like to maybe have information about 3 phytoplankton size classes, which is going also to give us some information about how the phytoplankton biodiversity is changing over time. And by using this prime production model, we will be able to relate better this information to the air-sea CO2 fluxes and the carbon export, for example. And finally, I'm just going to say that, as I said, I have obtained a fellowship from the French Space Agency to mostly here study the influence of a storm on the phytoplankton production in the Southern Ocean. So by using the framework that I presented, like the regionalization that I presented here, I will now be able to study within a region what is the influence of a storm on the phytoplankton production. So to see if this is inducing some anomalies in my average seasonal cycle that I've been showing you before. And so it has been shown by the past that the storm could have an influence on phytoplankton production, mostly when it's associated with some eddies and meander, and because the phytoplankton is able to respond quickly to this mixing induced by the storm. So that's why I would like to study for the next 2 years. And with that, I will stop, and thank you for listening to me.
Thank you, Nicolas.
Thank you.
Are there any questions?
Yes, Frank, go ahead. We cannot hear you if you are speaking.
Maybe in the chat?
I can ask one too while Frank sees if he can get
Yeah,
his audio worked out. Yeah, I was interested by your kind of pre-processing of the data before clustering with the PCA. I was curious if you tested out any other strategies for that before doing the Gaussian mixture modeling and how those worked.
Yeah, so I mostly focus here on using PCA analysis. So, but I tried different way of doing it and the numbers of principal component to use to see if I obtain different results at the end. But this is I obtain kind of always the same result at the end. So it's kind of really stable. It's like these features, but the seasonal cycle are very, I would say, very strong, and they always came up at the end. So whatever principal component or whatever, how I organize my data, if I from time, like I test a lot of things, and it's always showing up the same result. I also try with like because Raphael have been producing this 4D data quite recently because she have been using a new post-processing for the chlorophyll concentration, for example, that have been changed because we know that before there was kind of bias between the fluorescence and the real chlorophyll concentration. And this have changed just this year. And even when I'm switching from one data set to another, they always came up. So I will say that whatever filtering I'm using before running the Gaussian mixture model, I'm always obtaining the same regionalization. So yeah, I tried several of one and I always obtain this result.
Can you hear me now?
Yeah, I can hear you now, yeah.
Good, Nicolas. Very, very nice work, really impressive. So
Thank you.
I was wondering about the later slides. You had one where you showed weekly productivity estimates integrated with depth, and maybe I didn't catch the numbers right, but you showed something like 0.1 gram carbon per meter squared per week. And
yeah,
that seemed really low to me, but maybe that's what it is.
Yeah, so it's not primary production, it's a net biomass accumulation. So
Yeah.
I mean, it's
Well,
kind of
maybe
related.
that's just, yeah.
It's kind of related to primary production, but it's not exactly that. But yeah, and here I'm also like doing a big average for big region. So probably like if you take in one area, you might have higher values, but when you make an average of this big region,
It's still awfully low. I mean, even if it's a ballpark, it's.
yeah, no, I will expecting maybe to have like maybe one order of magnitude higher, but yeah, so that's why I still need to go through these numbers and check if like what is the range, like for example like the standard deviation around this value, because maybe it's whole hidden between inside that. But yeah, that's for the moment that's the value that I have for the moment. But yeah, I still need I just obtained this result last week, in fact, so I need to double check them.
Yeah. Thank you.
Hi, Monty.
Hi, Nicola. It was very, very interesting. My question is, how do you separate phytoplankton biomass, POC, and chlorophyll A? I'm not
Yes.
clear about it because not all POC is possible to attribute to phytoplankton biomass. Yeah.
So yeah, there's several. I mean, there's one way to do it is in fact is to look at the chlorophyll and the BBP profile together. And so when you have no more chlorophyll, you have to like kind of put the BBP at 0 or like some none. And so like this, you don't you remove kind of the POC, which is not associated with the phytoplankton community. That's how I have done it. Is that I suppress this information of BBP when I don't see any more chlorophyll associated with. That's one way of doing it, and that's how I use it.
Okay. Thank you. Yeah,
Sure.
it's very impressive. All the
Thank
best.
you.
Yeah.
Thank you.
Any other questions? I have one follow-up to my earlier one. Oh wait, we have one in the chat actually.
Oh.
Let's see. Bruno asks, what do you plan to use as a carbon fixation efficiency factor?
So it's a carbon efficiency factor for the primary production model, I guess. That's going to be the primary production model that have been developed here at the lab. So it was a machine production model that had been developed by André Morel in the 90s, in the early 90s. So that's the one that I would like to use first. But I'm also going to use also the primary productivity model that have been published, like the CBPM and the CAFE model, mostly from the American community. So that's the one that I would like also to apply. But first, I would like to apply the one that have been developed in the lab here. And also these parameters, the physiological or photosynthetic parameter that are available. Julie Abbott, with who I'm working, have also published some values for the different phytoplankton groups. So I would like to use those kind of value. And this value is also depth dependent. So I also would like to use those one.
Frank, go ahead.
Yeah, I was very curious about the long-term trends that you see, or no trends. You know, it's compared to what everybody says. You know, that there's big warming in the Southern Ocean, and I would have thought that this affects the mixed layer depth in some way. You know, and that would lead to some changes in the biomass. But so I was curious about that. If you see long-term changes in the mixed layer depth and in the warming in the different areas that you looked at.
So if you look at the mixed layer depth within this region, you can start to see a trend in terms of mixed layer depth. And because I have seen, as I was showing just around the end of my presentation, I was showing that there is a relationship between the mixed layer depth and the biomass accumulation. I will say that there's going to be some kind of impact over the long-term perspective on the biomass accumulation in this region. But in terms of phytoplankton phenologies, if we look at the seasonal cycle, I would say that the seasonal cycle is not going for the moment to change a lot. Like in every area, you are going to see the production occurring in January and February, but the values associated with this peak of production is going to change over time because of mixed layer depth. But in terms of like annual cycle, I don't think for the moment, but we've seen a clear change. I mean, probably some kind of like few degree latitude differences, but in terms of like uncertainty behind my analysis and the data, I don't think I can clearly see or say yet that we have a trend. But we might have one, but I'm not sure that I could say this with the data that I have right now.
Very interesting.
Thank you.
Jen, did you have another question? Or?
Oh, I was just going to ask another question about the methods and how much time you spent playing with the number of groups in the Gaussian mixture model and how you settled on 6.
So yeah, you can play a lot with it. In fact, it's still like it's I would say not random, but it's how to say it's based on the one that it's based on what I'm thinking should be the number. But still, I'm using some method behind to try to see if 6 is a good number, and for that I have these 2 criteria that I'm using for that. And they all show me that 6 could be a number, but in fact, you can also use a higher number like 8 or 12, but it's just that 8 or 12 is start to being difficult to interpret all of them and start to be, it doesn't bring me any, like I prefer to select 6 because like I have a small enough number to be able to have a way to better group my data and have a better summarizing information. But yeah, it's clear that when you play with Gaussian mixture model, you can also play with the number of groups. So I choose 6, but maybe someone else will choose something different if you are interested. If you are interested maybe in some area, like I don't know, I don't know, if you're interested by, for example, in the Indian Ocean, maybe you would have said like, oh, I need another region because I'm interested about this particular region. But in my case, and for the global Southern Ocean, I think 6 is a good number. But yeah, I have some criteria to to support this choice as well.
Got it. Thanks. And just to point out, Frank mentioned that it would be interesting to compare your classification to some other seascapes developed from satellite data.
Cool. Yeah, sure. Don't hesitate to contact me by emails or whatsoever. I will also be in Vienna for the ones that are going to Vienna. So if you want to have a chat in Vienna with me, I will be there the whole week. But otherwise, yeah, just shoot me an email and I will be able to compare. This is not a published work yet, so I hope I will be able to publish this quickly. I mean, quickly, I don't know how quick it can be, but but I would like to publish this, and so probably I hope over the year I will be able to publish this information.
Okay, so I think there is no more questions.
Good. Well, thanks again.
Thank you, Nicolas. Thank you very much
Thanks,
for your
Nico.
presentation.
And thanks everyone for joining.
Bye.